Cool. All right. Welcome to the second lesson for week number 6, task number 2. We are looking at meiosis, and just like we did with mitosis, we're looking at its relationship to the body or in the body. With the success criteria, you should be able to describe and explain the process of meiosis. And then that second bit there where you're talking about greater variation than asexual reproduction, that's going to be our final task for this week. So it's going to be basically a 2-part lesson. Cool. But before I go into that, let's kind of give some background context to why we need meiosis. So first off, when we think about sexual reproduction, the only source of variation, if you guys remember me saying in the last lesson, was mutation. Sexual reproduction is going to give us that huge advantage because it's going to create more variation sources, and that ties into meiosis. The other thing about sexual reproduction that is more beneficial than asexual reproduction is that you have 2 parents, and so with the 2 parents, you then are getting a blend of the genetics from each of them, thus making the offspring different from the parents. Useful, because if you guys remember in our first lesson, we talked about the benefits of variation and how if you have more variation in your species, in your population, if there's any event that's going to result in any death, then you're more likely to survive it. Cool. And in order to obviously prepare for having 2 parents, we need to half the DNA in each parent, so that way you restore the chromosome numbers when fertilization occurs. If we didn't do that, the chromosome numbers would keep doubling with each generation, and that can cause problems. Interesting though, plants do that. Plants are weird. They can operate with a strange number of chromosomes and be fine. All right, so meiosis. So meiosis is a very specific type of cell division. It is used to produce gametes, which are sex cells, eggs and sperm, and it's needed for sexual reproduction. That is the only role meiosis plays in our body. When it does this, it's going to produce 4 cells. These cells will have half the chromosome numbers of the parent cell. Thus why we call it a haploid. I like to remember it as they're both starting with H, so haploid, half. Takes place in the ovaries and testes, aka the sex organs. It produces variation, so it's really, really useful. This is an old-timey image of what they thought the sperm was, and they thought like the sperm was what developed into the human. They didn't quite realize that it's the sperm and the egg. It's just interesting to see what they used to think. Anyway, let's go into meiosis. Now, meiosis is a little bit more complicated because there's 2 steps to it, meiosis 1 and meiosis 2. So the nice thing is there is going to be some similarities. First off, pre-meiosis, nucleus, genetic material, number of cells 1, genetic material, we're going to go 2x, and I'm going to write 46 again. I think it's helpful to think of that in context of humans. All right. The second step is also the same. We have to replicate the DNA. So I'm going to draw this as more concentrated. Still 1 cell. Now we have double the amount of DNA that we need. Again, the steps are all very similar at this point. The nucleus is going to start to degrade, and my DNA is going to start concentrating down. Now, I draw 4 chromosomes, that way you can kind of see the process of what's going on. Cool. 1 cell, twice as much information. There we go. All right, so here's where the first step now differentiates from mitosis. In this case, instead of the chromosomes going into a single file line, they're going to go into 2 lines. When they go into those 2 lines, they're actually going to buddy up with their homologous pair. So remember how I said you got 1 chromosome from mom and 1 chromosome from dad. The reason why that happens, or why, the reason why you get 2 sets of DNA. So this is basically it kind of aligning in their kind of square dance pair lines. So there we go. And the spindle fibers again. What was I going to say about this? Homologous pairs, yes. So with those homologous pairs, what a homologous pair is are chromosomes that are the same size with the same genes. And I think I do have a slide in here. Yes, I do. Showing homologous pairs. Now, in this case, this is the unreplicated chromosome. So do keep that in mind. And this is supposed to show 1 chromosome from mom, 1 from dad, 1 from mom, 1 from dad, etc., etc. So with those homologous chromosomes, they're obviously the same length, but they also have the same genes and the same order, the same pattern, the same staining pattern when we stain it for the various genes. The specific gene location is known as a locus, just FYI if you see that word come up. And when I do something like this and take a picture, what we're looking at then is a karyotype. So that is basically looking and seeing how many pairs of chromosomes we have. So we have 23 pairs, so 46 chromosomes in humans. And that's what we're seeing with our karyotype. And when you look at other animals, that's what they're referring to with their chromosome number. Yeah, that's all I wanted to say for now. I think I'll pause here and I'll show you my 23andMe, because that's really cool to see. All right, cool. Just a little detour into my 23andMe because I think it's really neat. My brother got this for me as one of my Christmas presents, and it is the most fascinating thing. And also, like, if you're a science nerd like me, it's just great information. And also, I find it's really useful teaching-wise. There's lots of information on the 23andMe, but the main thing I wanted to show you is my chromosomes. As you guys know, I'm biracial. My mom's Chinese and my dad is European, or Paki, I should say. And because I am biracial and both my parents are, full white and full Asian, I have a really clean look at my chromosomes. So with these chromosomes here, you can see the 23 pairs, and you can also see that I've inherited 1 from my mom and 1 from my dad. The things that are obviously orange and red are coming from my mom's side, and the things that are blue are coming from my dad's side, which is the European side. So it's a really nice kind of clean cut because you can see basically each chromosome that I got from my parents. I'll talk more about this in our next lesson and how what my eggs would be like. Because again, like you can visually see what's going on. All right, cool. Let's go back to this. So, like I said, the homologous pairs will line up and they will split apart. So in this case, we're not getting that splitting of the sister chromatids. The whole chromosome is going. So there's no splitting or breaking apart in this case. And then those are our dots to break the cell apart. Cool. So that's what that looks like. We have 1 cell still because there's not that pinch off of the material, and still technically all the material is still within the cell. And then they will split apart. Now, in this case, I have just drawn it by chance that the 2 red ones are going together and the 2 blue ones are going together, just to kind of highlight the fact that these cells are different from the parents. So I have 2 cells. Genetic material-wise, I have 4 times still, 92, but in each cell, it's 2 times, so 42. I want to make a special kind of point here that even though this is the normal number of chromosomes again, sorry, let's say 46, these aren't functioning cells. These are just the intermediate step to producing our sperm and egg. So do kind of keep that in mind. All right, so that's the first cell division. The second cell division then, same thing happens. The chromosomes start to concentrate, and the nucleus starts to degrade away. So now we're entering this mitosis too. So we have two cells. We still have 4x 92 chromosomes, but it's in reality 46 per cell, just to kind of keep it all straight in your head. All right, now the chromosomes will line up single file. This is going to start looking like what we saw in the second half of mitosis. But the key difference here is that we have divvied up the chromosomes. So pull it apart, pull it apart. Cool. 2 cells, 4x, 92, 46 per cell. All right, then they start to split apart. And so again, this is going to be when the sister chromatids pull. And the cell membrane starts to form. So a lot of this is just going to be repeated, just to kind of keep track of what's happening. And then again, final step, nucleus forms again. In this case, we have even less DNA inside there. That's because it's been divided. Cool. So number of cells that are being produced is 4. Genetic material-wise, now when we're thinking about per cell, it's just X, so like 1X. So 23 chromosomes if we're thinking about humans. So we have halved the number. That's why we're calling it a haploid, is the name of the cell. And that's because when fertilization occurs, 2 cells are going to come back together and they're going to restore that number back to 46. Some interesting things to note with the difference between sperm and egg, at least in the human sense, and probably some other organisms as well. When it comes to creating sperm, the cell division is even, and you're going to create 4 sperm. When it comes to females, the cell division actually isn't even, and what you get are things called, I believe they're polar bodies. They still have the DNA, but one of the cells in that cell division gets more of the organelles and cytoplasm and basically just the resources of the cell. So you get one real chunky sex cell, and then the other 3 are really kind of defective smaller cells. And the reason why is because that egg needs to have a lot of those nutrients, because they're going to jumpstart and make basically provide all the components outside of the DNA from the father to the new cell. So just to kind of keep in mind, the only thing that's in that sperm that contributes to the offspring is that DNA. Everything else comes from the egg, from the female. So one of the things you might heard of things being referred to is the mitochondria, which we'll cover in another topic. And that the mitochondria is the powerhouse of the cell. It produces all of our energy. The interesting thing about mitochondria is that they have their own DNA. And because you only get your mitochondria from your mother, that mitochondrial DNA can trace back the maternal line. So that's a cool kind of thing to analyze. All right. Cool. here's our little joke. So meiosis gets kind of thrown off because you think that they're going to stay with their sister chromatids, but then they don't in anaphase 2, which is during that second meiosis cell division. The other thing I like to mention is what if there's a mistake? So if we look at our cell division steps, we have 2 division processes. We have a division process here, and we also have the sister chromatids splitting here as well. Those things can occasionally not split properly. And basically what's going to happen then in those daughter cells is that one of the daughter cells will have too many chromosomes and the other daughter cell will have too few of them. And again, like I was saying, it's really important that we have these chromosome numbers, because if we double with each generation, we get problems. And even if there's just one extra chromosome, it can cause a huge amount of potential downstream genetic issues. The example that I use is Down syndrome. So you have an extra chromosome number 21. In other cases, it might be really minor and it might be unnoticeable. You can get extra Y chromosomes, and it doesn't seem to cause any issues. But to be fair, the Y chromosome doesn't have a lot of genes on there. So it's just something to kind of note. In some cases, the extra chromosome or the absence of a chromosome will lead to an inviable zygote or fetus. And other times it will carry to term and actually survive well into adulthood. So it really kind of just depends on what chromosome and what genes it affects. Oh, and here's a fun fact. So all tortoiseshell cats are females. Here's my kitty cat. And I should say the thing that's characteristic about tortoiseshells is that they have patches of black fur and patches of orange fur. The reason why is because the color coat for cats is located on the X chromosome. Now, in females, we obviously get 2 X chromosomes. Males only get 1. So females actually inactivate one of the X chromosomes because they don't need both to function. And it ties back to when we were talking about the sex-linked alleles and how males are more likely to be affected by X chromosome mutations than females because we can inactivate one of them. We get 2 chances of getting a normal allele. Anyway, since the tortoiseshell inactivates one of the chromosomes, it means that you get that random patterning of black and orange. And this happens during fetal development. Now, you can actually get male tortoiseshell cats, but this only happens when you have that error in the chromosome division, and the male cat ends up with an XXY. So it's still male because it has the X, or it's still male because it has the Y, but it has 2 X chromosomes, so it's going to do the same thing of inactivating one of them. So you can get male tortoiseshells, but they're extremely rare, and because they are so rare, they're considered lucky. So just a fun fact for you guys about tortoiseshell cats. Cool. The other thing that we need to think about as species is looking at the ability to produce fertile offspring. So when we classify something as a species, it means that they can mate and produce fertile offspring. One of the big factors in deciding, the ability to produce offspring will be that chromosome number. So basically what we're thinking about is, why can't we cross species together? Why can't we cross a cat and a dog together and get like the super pet? It has to do with chromosome numbers is the main reason. The other reason obviously is the larger genetic component. But chromosome number is a big factor into why things don't happen. So we can't have any like centaurs because if you look at the chromosome number for humans versus horses, they're drastically different. So sorry, not possible. I was actually asked that question once when I was a teacher or when I was working at my last school. Cool. Okay, so Meiosis Recap. And this is, again, a good comparison to what we were dealing with previously with mitosis. In this case, we're looking at meiosis. So where does it occur? It occurs in the sex organs. So testes and ovaries. Number of divisions, in this case, there are 2. So we have 2 cellular divisions. That's why we have meiosis 1 and meiosis 2. It creates 4 cells. The number of chromosomes is going to be halved. Types of cells it creates, my bad, is the gametes, which are your sex cells. I think that's spelled with an A, not an E. Number of cells it produces is 4, and the role in the body is sexual reproduction. Cool. All right. So that's a little recap for meiosis. So by the end of today's lesson, you should know what meiosis is and the role it has in the body. You should be able to describe and explain that process of meiosis. This you will need to do in this standard. So do kind of keep that in mind, that understanding meiosis is important for this, because we're talking about genetic variation, and meiosis is a great way to produce that genetic variation. Cool. So now it's time for you guys to go onto your Mahi choice. All the stuff is listed on the Google site, and the resources are all on Google Classroom.